Welcome, everyone. We often talk about DNA as the blueprint of life, but a blueprint is just a silent plan sitting on a shelf. To actually build a living, breathing organism, the code has to find its own voice. Today, we'll be diving into the world of eukaryotic transcription, the sophisticated molecular machinery that reads the genetic code and transforms it into a message. We are moving from the stillness of the double helix to the dynamic world of RNA. In the next few minutes, we'll be looking inside the nucleus to see how the cells decide which genes to turn on, how they avoid mistakes, and why this specific process is the foundation for everything from your heartbeat to your immune system. Let's unlock the code. Close your eyes for a minute and imagine that you're in the most secure library ever built. No noise, no crowd, only knowledge. Inside this library lies the instruction manual for you, your height, your heartbeat, and even how your cells know when to grow and when to rest. This is your manual. This manual is your DNA. But here's the rule. The book never leaves the library. So how does the cell read these instructions and use them? How does information travel from the DNA to the life itself? The answer is a beautiful, carefully controlled process called transcription. So today let's step inside the nucleus and watch how the life's code gets copied safely, precisely, and elegantly. In eukaryotic cells, which include humans, plants, and animals, transcription happens inside a special compartment called the nucleus. Think of nucleus as the VIP room. DNA stays protected here at all times. Transcription is the process of copying information from DNA into RNA. Basically, you make a copy of RNA from a specific segment of DNA. That is what transcription is. So it's like making a photocopy of an important recipe. So not the original. So the original stays safe while the copy can be used outside. It's the foundational process and the fundamental process in all living cells, and it occurs in both prokaryotes and eukaryotes, though the mechanisms differ. It's the most critical part in central dogma as well. Before we move on to the transcription, before transcription can even begin, we need to meet the main workers, the key players in transcription. First is the RNA polymerases. So in eukaryotic transcription, there are three RNA polymerases that work each have a specific role to perform. Fine. So the first RNA polymerase 1, makes the ribosomal RNA. It makes the structural core of ribosomes. RNA polymerase 3 makes the transfer RNA and small RNAs which are used during translation. And the most important, the star of today, is the RNA polymerase 2, which transcribes messenger RNA, which is the molecule important for carrying the genetic information from the DNA to make proteins. So it basically handles the genes that become proteins. Okay. So because proteins control the structure, function, and regulation, RNA polymerase 2 plays a very important role in the life of the cell. The next key factor or key players is the transcription factors. Now, unlike the bacterial RNA polymerase 2 cannot start transcription on its own. It requires help, and this help comes from a group of proteins called general transcription factors. These factors are important because they recognize the correct gene, they mark the exact starting point, they position the RNA polymerase 2 properly, and they prepare the DNA for opening. Without the general transcription factors, transcription will become inaccurate and chaotic. Together with the RNA polymerase 2, they form a structure which is called the pre-initiation complex, which is the foundation for transcription. Now let's meet this team briefly and simply. TF2D, which is the identifier, everything begins with TF2D. TF2D contains TBP, the tata binding protein, which recognizes and binds to the promoter. This is the moment the gene is officially identified. Without TF2D, the transcription machinery cannot assemble. TF2A and B, the next are the positioners. So next comes TF2A and TF2B. They stabilize the TF2D, which is on the DNA, and they help them to stabilize stabilize the TF2D on the DNA and help to define where the transcription is going to start. They make sure that the machinery is aligned, not too early, not too late. TF2F. TF2F is the escort. It arrives carrying the RNA polymerase II. It helps polymerase to bind correctly and prevents it from attaching to the wrong DNA regions. Now the enzyme is finally at the right place. TF2E and TF2H are the activators. TF2H plays a very important role. It uses its energy to unwind the DNA and it phosphorylates the RNA polymerase II. This is a chemical modification. It's like a green signal. So that says the transcription can now begin. Now the third important key player is the promoter. So where does all this assembly happen? It happens at a region called promoter. A promoter is a DNA sequence located just upstream of a gene. It does not code for protein. Instead, it gives instructions about where and how transcription should start. One of the most well-known promoter element is the data box. The data box acts like a clear start line telling the transcription machinery this is the correct gene. It says start copying from here. Once the transcription starts, RNA polymerase II just moves forward, has TF2H phosphorylates the polymerase. RNA polymerase II clears the promoter, which is a step called as promoter escape. At this point, the transcription factors remain behind. RNA polymerase II moves into elongation and the gene is now actively transcribed. This transition makes the shift from preparation to action. Before we go deep into before we go deep, let's take a step back and see how big the picture is actually. No matter how complex transcription seems, it always starts with three main stages. First is initiation, where the gene is selected, the machinery assembles, and transcription begins. Next is elongation, where the RNA polymerase moves along the DNA, copies the genetic message, and finally there is termination, where the message is completed and released. If you understand these three steps, you understand the backbone of transcription. Now let's go through them one by one, starting with initiation. Now we enter the first active stage of transcription, initiation. Before anything can be copied, DNA must be assembled. It must be assembled. Okay, it should be accessible. Fine. So in eukaryotic cells, DNA is wrapped tight around proteins called histones. They form chromatin. So the first challenge is simple but crucial. The gene must be uncovered. Special proteins called chromatin remodeling complexes. They loosen up the DNA from the histones, opens this region just enough for the transcription to start. This step decides which gene is allowed to be read. Once the DNA is accessible, the general transcription factors assemble at the promoter along with the RNA polymerase II. They form together the pre-initiation complex or PIC. The pre-initiation complex, it acts like a carefully built launch pad. It ensures the correct gene is chosen, the correct start site is marked, and the machine is properly aligned. With the PIC pre-initiation complex in place, the double helix starts to unwind at the start site. A small region will start opening up, and this region is called transcription bubble. For the very first time, the DNA bases are exposed and RNA polymerase II starts to read the template strand. Okay, so now initiation is complete and the gene is now awake. Now next step is elongation, the main copying phase. RNA polymerase II moves along the DNA smoothly, like a train moving on a rail track. As it reads the DNA, it builds a complementary RNA strand. This is a very important rule to remember. In RNA, uracil replaces thymine. So that is U replaces T. So whenever DNA shows a RNA adds U. This small change clearly marks the molecule as RNA and not DNA. Okay. So this is important. This is the Step 1, it starts synthesizing RNA strand. It synthesizes from 5' to 3' direction. Every message needs a proper ending. So when the RNA polymerase II transcribes a special signal called the polyadenylation signal, the RNA is cut and it is released. But the polymerase doesn't stop immediately. A molecular machine acts like a torpedo, chasing the polymerase and knocking it off from the DNA. Only then does the transcription fully stop. The DNA rewinds and it is protected once again. Right? This is the end termination. Now, once the RNA molecule has been produced during transcription, it is called as a pre-mRNA, and it is not ready to leave the nucleus yet. Think of it as a rough draft. Before this message can be used to make protein, it has to be processed, protected, and edited. This entire preparation step is called RNA processing. First is 5' capping, protecting the first modification happens at the 5' end of the RNA. A special chemical structure called 5' cap is added. Why is it important? It protects the RNA from being broken down. It helps the RNA exit the nucleus, and it helps the ribosomes to recognize the mRNA during translation. You can think 5' cap as like a helmet. Without it, the RNA will become unstable and easily destroyed. Second is the poly-A tail or polyadenylation tail. It helps. Next is a long chain of adenine nucleotides which are added to the 3' end of the RNA. This is called poly-A tail. The poly-A tail is important. It increases the RNA stability, prevents degradation, and helps to control how long the RNA survives in the cytoplasm. The longer the tail is, the longer the message can be used. Together, the 5' cap and the poly-A tail acts like a protective book, covering it and keeping the message intact. Third is splicing. Splicing once it comes out, one of the most important steps. Okay, now comes one of the most important steps is splicing. Eukaryotic genes are not continuous. They have introns, which are non-coding regions, and exons, which are coding regions. So during splicing, the introns are removed and the exons will be joined together, forming a meaningful message. This editing process is done by some complex molecule machine called spliceosome, which are proteins basically. Without splicing, the RNA would become unnecessary information and it will produce faulty proteins. So in order to have a proper and meaningful message, the unnecessary informations are removed. Okay, that is what splicing is. Okay, so you have you have alternative splicing, which is where the eukaryotic cells become truly powerful. Through alternative splicing, cells can join the exons in different combination. This means one gene can produce multiple mRNA molecules and therefore multiple proteins. This is how humans create enormous protein diversity from a limited number of genes. Alternative splicing allows cells to adapt, specialize, and respond to different needs, all using the same DNA. Now let's pause and compare how transcription works in prokaryotes and eukaryotes. In prokaryotes, everything is fast and simple. There is no nucleus, only only there is one RNA polymerase, and transcription starts quickly with the help of sigma factors. There is no processing needed. The RNA is ready to be translated almost immediately. Eukaryotes, on the other hand, it is a more careful and controlled approach. Transcription happens inside a protected nucleus. It uses three different RNA polymerases and requires multiple transcription factors. The RNA is capped, tailed, and spliced before it can be used. So while prokaryotes focus on speed, eukaryotes focus on precision and regulation, which is a key reason complex life is possible. So prokaryotes are fast, they are simple, and eukaryotes are slower but highly controlled. Same goals but different strategies. I hope this journey into the nucleus has helped you appreciate the incredible molecular teamwork that happens inside your cell right now. Now, if this video made transcription clearer, share it with someone who loves biology too. Until next time, keep questioning, keep learning, and keep exploring the language of life. Bye.